welcome everybody that has joined us. This is Best's second webinar that we've actually hosted. For everyone, just to let you know too, that we will be recording this webinar and posting it to all registrants. So anyone that has registered, we will actually put it up on our social media posts and send you a link to them at the end of in the next few days. I just want to quickly introduce the panelists. Kieran Knight is an agronomist for Pest. So Kieran's here to help with the answering of the questions at the end of the webinar. Joel Williams obviously is who we're all here to see. So I'll let Luke sort of introduce Joel in a more detailed way in a second. But also we are hosting this webinar with the help of Luke Harrington from Regen Farming. So I'll introduce Luke to you guys. He can sort of introduce himself and talk a bit about there. On the housekeeping side of things, feel free to send us a chat, anyone that wants to. Just maybe let us know whereabouts you're listening in from. We're sort of all keen to know how widespread the listeners go. So please feel free to put that in. And on that note, Luke, I'm going to pass over to you to introduce Joel officially, and let's get it started.
Yeah, no worries, Darren. Thanks for that, and thanks to everybody for joining us today. Just really quickly, I met Joel probably a bit under a year ago in Wagga. He was giving a talk on cover cropping, and I went over there to listen to him because I was going to poach him to do our Simple Soil Health Solutions day that we normally have in Deniliquin, and that normally runs about now. But unfortunately with COVID and all things happening, that hasn't happened. So we thought we'd get Joel to come over here. And I was really impressed with the way he came across and how he spoke and his depth of knowledge and what he was talking about. So I think today will be a very interesting day. And yeah, I think it's a really topical question at the moment with the amount of things that have been used lately. And this year it's been planes going everywhere with fungicides and insecticides and things like that. So I think, yeah, we really should be looking more at our at nutrition and soil health and biology for our IPMs. And moving away from these other things that we're using, the insecticides and fungicides, because those tools I think are going to get taken away from us in the next decade or so. We're going to lose some of those through different things. Yeah. So anyway, I'll hand you across to Joel, and yeah. And the other thing too, I'd like to ask is any questions, if you can put them in the Q&A box, not in the chat box, because it's really difficult to read them and keep up with them if they're in the chat. If they go in the Q&A, we'll be able to answer them at the end of the day. So, alrighty, thanks for that. And over to you, Joel.
Awesome. Thank you very much, Luke and Darren, and pleasure to be here. So let me just share my screen. Okay, is that working?
Yep.
Yeah. Okay, so thanks again. And the topic for today is then integrating nutrition and biology for pest and disease management. So there's my website and Twitter handle there for anybody who might be interested. So the way I view integrated pest management is, in this context, is a combination of these aspects. And we're going to talk particularly around nutrition, biology, and a little bit around plant management as well. So here I'm referring to optimizing the nutritional balance in the plant, the soil or the plant, but particularly a focus on managing certain nutrients, key disease fighting kind of nutrients. Biology, we're talking about the living component of soils, particularly microbes. They can offer all sorts of protection against disease, and both microbes as well as some of the larger soil insects and things have a role in helping to manage insect pests as well. So we want healthy, biologically active soils, but also there are certain species that are particularly, microbial species that are particularly antagonistic. So we'll touch on those. And really what nutrition and biological interactions do is they help to support plant immunity. they help to induce a resistance, a systemic immune response in the plant. And so that's kind of what we're going to talk about, and make these two topics practical and say, well, how do we manage these to optimize plant immunity? So that's going to be the bulk of our discussion. And I'm just going to say a few quick words. I've just got a couple of slides just to talk about a little bit on plant species diversity and how little steps away from some of our monocultures can also just be a part of that puzzle. We could have a big discussion on that one, but it's not the focus of today. We're really going to focus heavily on the top two, as the title says. So that's kind of the outline for today. So if we talk about the concept of plant immunity, for me, it comes back to photosynthesis and having the right nutrients that catalyze photosynthesis. And it is photosynthesis that then fuels the production of all sorts of defense chemicals. It fuels the production of the plant's immune system. So we do have to kind of come back to a basic fundamental chat around nutrition and photosynthesis, and then we'll kind of link that to specific some of these immunity topics. So plants, they do have an immune system just like us. Their first line of immune system, their first line of defense is a physical defense, toughening up their skin. Like us, the plants, also their skin is their first barrier to the outside world. So that's called a passive defense. That's a physical strengthening of the skin of the plant. But then internally, they also can produce a range of biochemicals that have antifungal, antibiotic, or insect repellent, or feeding deterrent kind of chemicals against insects, which these are products of photosynthesis, which they can also synthesize. So this is more of a biochemical response. This is an internal range of biochemicals that have these antagonistic properties against insects or disease organisms. So if we talk about, firstly, say, passive defense, here I'm talking about a physical barrier, a reinforcement, a strengthening of the plant's cells, the cell walls, that skin of the plant. But it goes beyond that. You know, we could talk about things like spines or hairs, thorns, these kinds of structural things, hairs that can trap insects and prevent insects from getting to the plant cell. So there's physical barriers first. And then, you know, going a little deeper, we can think about some of these kind of cell wall and layers of the plant's skin. And you can see this kind of list of things, various, you know, under the cuticle there, we've got various waxes and cellulose and pectins and all of these various structural compounds. And these structural compounds also help to reinforce those cells and making the plant a more robust structure, thereby making it more difficult for, say, an insect to chew through this skin or a disease to penetrate through this skin. You know, if we think about, for example, a bacterial disease, it might be the size of just one of these little dots here. I mean, it's got a long way to penetrate through to cause disease in those cells. So the more proactive we can be about this first line of defense is a very important and proactive step. So that's your passive defense. Then we have more of these active or internal biochemicals that the plant can produce. And these are typically systemic chemicals. They can be translocated throughout the plant. And this involves all sorts of signaling responses where when an insect has a chew on a leaf, for example, or when a pathogen begins to start to cause that infection, it sends all sorts of signals throughout the plant. And the plant will respond to that attack by recruiting microbes from the soil, taking up additional nutrition from the soil, and then again through photosynthesis producing a range of these defense chemicals, these very often very targeted, very specific chemicals that can kill or deter or block an invading pest. So there are two aspects to plant immunity that we'll touch on regularly through the rest of the talk. And so again, as I say, it comes down, however, to photosynthesis. The plant producing these structural compounds, producing these biochemicals, these internal systemic chemicals, it all takes and requires energy. And these compounds are all products of photosynthesis. So, you know, it comes down to managing this fundamental equation, the plant taking in Yes, photosynthesis initially is about producing glucose, that simple sugar kind of molecule, but that is the building block in which the plant will then produce a whole range of other compounds that it requires. So it's breathing in that carbon dioxide, taking out water, then we have carbon, hydrogen, and oxygen, the building blocks to build plant biomass, but also to fuel this immune response that I speak of. So it comes down to this fundamental equation, but it's not just about sun, about carbon dioxide, and about water. It's also about the other key ingredient is nutrients, and that's the soil fertility part. The plant requires macro and micro minerals as catalysts to drive that photosynthetic process. We need the macro and micro minerals. They act as catalysts in supporting the synthesis of photosynthetic products and ultimate plant growth. So that's a nice, very simple equation. If we just take that simplicity and kind of add a little more detail, I'm saying the exact same thing here, just with a little more complexity, a little more detail in the message here, but it's saying the same thing. You know, it's carbon dioxide, it's water. In the presence of sunlight, again, the plant requires minerals that act as part of enzymes or catalysts that help to build or synthesize sugar, plus, of course, oxygen as a byproduct. Now, the point is to say, if we have a mineral limitation here, then this equation will not flow as optimum. Okay, we will not be optimizing this process if we have mineral deficiencies. Now, once the plant builds that first little glucose molecule, as I mentioned before, it uses this as a building block, and it begins to stitch lots of sugar molecules together into longer chain sugars, larger carbohydrates, etc. So, and again, we require minerals for this second step. So we're now taking the simplicity, the building block, and turning it into diversity and complexity, more of these various other compounds. So from complex sugars and carbohydrates, the plant can also link in some nitrogen or sulfur into the carbon, building amino acids, linking those together, building proteins. The plant can also build various fats, oils, waxes, these kinds of compounds, and the list goes on and on. Hormones, vitamins, aromatic compounds for smells and scents, these volatile compounds, various colors, phytonutrients, flavors, things that are good for us, nice flavors, antioxidants, protective chemicals, protective compounds, defense chemicals, and indeed rutixidates. So it's to say that then this second step, all of these other products, these more diverse and complex compounds, these also are photosynthetic products. They also require macro and micro minerals as catalysts to convert that first little building block into these more diverse and complex various structural and immune enhancing compounds. So when we talk about plant immunity, we are really talking about this second step. It's these various complex compounds that are the real quality enhancing, but also immune enhancing. And so again, the simple point is to say, if we have a limitation of any one of the essential minerals, essential plant nutrients here or here, then either of these processes will not flow and function adequately, and therefore immunity will be compromised. And so that's really to say that if you have a limitation in any one of these essential minerals, and you know, I'm not going to go through this list in detail today, you know, they have all sorts of various functions in which these minerals help the plant perform. If we have a limitation in any one of these, ultimately in one way or another, yes, we're going to be limiting plant growth, plant production, plant biomass, but also we'll be limiting those other biochemical pathways that help to produce those immune enhancing compounds and structural compounds. So they're one and the same. If we want to grow, you know, I know many of the listeners will be interested in how to produce more yield. If you want to do that, your process of doing that is the same as enhancing immunity. In many regards, there's a lot of overlap. If you have a limitation of anything, it will limit plant production, but that will often, in many occasions, link to some other immune pathway. So you'll also be limiting plant immunity in one way or another. If any one of these functions you see on screen here are not performing adequately, plant health will be compromised. So beyond that, in that simplified comment to say that a limitation of any nutrient, which is true, will limit immunity, we can then move in to say, okay, what about certain and specific nutrients that are well-known disease fighters or particularly immune-enhancing supporting minerals? So here's a bit of a list of some of the top candidates. So, for example, things like calcium, silicon, and boron. All three of these minerals are deposited in the cell walls. They work synergistically together and they help to reinforce those cell walls, improving that structural robustness of the cells and therefore ultimately of that plant skin. So those three have a very strong link to cell wall strength. And there's a lot of good evidence that shows that, you know, that therefore the stronger the cell walls, the more difficult it is for insects to chew through, the more difficult it is for pathogens to penetrate through. So if I were wanting to be proactive about pest and disease management, I would ensure that I am optimizing the availability of calcium, silicon, and boron, and making sure that I have adequate levels of those minerals in the plant tissue. A few others, manganese, copper, and boron again. These three trace minerals all have a role, particularly in producing some of those structural compounds that I showed at the very beginning, toughening up the skin of the plant. So things like cellulose or lignans, hemicellulose, some of these other pectins, waxes, these kinds of other carbon-based kind of compounds. Those three in particular have a strong link in building those kind of structural compounds. So again, I would be proactive about monitoring and managing my manganese and copper and boron. Again, those first two on that list, the calcium, silicon, boron, manganese, and copper, those first five, I should say, the first two bullet points, those are the ones that are particularly important, again, for these skin of the plant, the primary defenses, that first line of defense. And when we move into then discussing the immunity from within, that internal immunity, silicon and manganese emerge as two very important minerals in driving the production of these immune chemicals. Okay, so that's kind of the top list there, saying that yes, a deficiency of any mineral can compromise plant health. So address any limitation you have, but I would be, if you have a particular history or problem of disease or insect pressure, I would be zoning in on those particular key disease-fighting minerals. And then at the bottom there, I've got excess nitrogen, and I will come back and just say a few quick slides on this one. It's just to say that when we push nitrogen too much, too high, as much as that can push growth, that growth will often be weak, spindly growth, not robust plant tissue. And again, that is inherently making the plant automatically weaker, more prone to pest and disease attack as well. So nitrogen is critical and essential mineral, but when we over apply nitrogen, we can push too much kind of lanky growth that's not supported. And really the crux of that problem really stems down to that nitrogen doesn't work in isolation. It requires other minerals to work with it in order for it to do what it does. And so particularly, I'll go through some examples in a second, trace minerals is a big one, that they work synergistically with nitrogen. So all I'm really saying is if we focus on nitrogen and some of the other supporting minerals, like trace minerals, and keep them in balance with that nitrogen, then nitrogen is much less harmful to plant health. Then it can be very supportive of plant health. When we see amino acid and protein synthesis, that's when nitrogen can enhance plant immunity. When we have free nitrogen, nitrates, free ammonium, urea, that's what the plant doesn't want. It wants amino acids, it wants complete proteins. So excess nitrogen, I'll come back to that one. So this is just to say that there's actually quite a lot of work out there that has explored the links between nutrition and pest and disease resistance. And I just put this one up there as an example, a nice review looking at the role of nutrients in disease in this particular example. And just to show you that there is a good body of evidence out there supporting some of the ideas that we'll touch on today. I'm just going to read a few highlighted points just from this abstract of this study. The control of plant diseases using classical pesticides raises serious concerns about food safety, environmental quality, and pesticide resistance, which have dictated the need for alternative pest management techniques. In particular, nutrients could affect the disease tolerance or resistance of plants to pathogens. When there is a high level of nitrogen, there is an increase of severity infection. Potassium can also help to decrease the susceptibility of the host plants. Manganese, as I mentioned, can control a number of diseases, and manganese has an important role, again, in that lignin biosynthesis and these other structural compounds, these phenols, and photosynthesis generally. Boron is another one, found to reduce the severity of many diseases because of boron, its impact on the cell wall structure, as I mentioned, and silicon as well, shown to control a number of diseases. And it is believed that silicon creates a physical barrier that can restrict fungal hyphal penetration or induce an accumulation, that internal response of many of those antifungal compounds. So there's studies out there that have looked at this nutritional link, and there's a lot of good evidence out there to support some of these ideas. If anyone likes some additional reading material, this one's pretty heavy going, but it's a very comprehensive mineral nutrition and plant disease, a really thorough book that explores this topic specifically. It goes through a whole host of different diseases and many of the mineral and nutritional links that have been identified, whereby either a deficiency or an excess of the mineral can lead to a greater susceptibility of the host plant to disease. So there's, just to show there is some reading material out there. And just to say then that there's, with good reason, emerging interest around the role of kind of foliar sprays, therefore then to deliver those essential minerals to help prime and support plant immunity with a nutritional approach. And the conversation of today is not about foliar sprays. That's a whole nother big discussion in itself. But I just want to put this study up there to highlight that there's a lot of knowledge gaps and that there's a lot of potential in using nutrition to combat disease. But we've really got to have the research, and we've got to have the right research agenda in order to look for these kind of examples and study these factors in order to help us use nutrition as a central tool for plant immunity. So here's just one example, just for interest's sake: foliar spraying of manganese on enhancing cucumber resistance against disease. So again, I'll just read a few key points. Foliar application of manganese is an effective factor in enhancing plant resistance to fungal diseases, but the efficacy of this method strongly depends on the application time. The excess levels of manganese, so adequate manganese, led to a significant reduction in fungal disease. The highest reduction in disease severity in this study was observed when manganese solutions were applied 4 days before the infection. So they intentionally infected the cucumber plants with disease and then sprayed manganese prior to that infection. So they found the optimum time of manganese spray was 4 days prior to infection. The cucumber plants treated by manganese at that 4 days days before the infection accumulated the highest level of manganese in their leaves than those treated at 14, 10, or 7 days before infection. Okay, so that's I know a very specific and horticultural example, but just highlighting that the timing of that application was also central in influencing the effectiveness of a simple manganese sulfate foliar spray in this instance. So that's just a little interesting example to show that there's a lot of opportunities, a lot of knowledge gaps, but there's a good evidence base to this concept. But certainly I'll admit there's plenty of knowledge gaps that we need to work on. And I threw this one in there also just for interest's sake on foliar sprays to say that moving forward, I think foliar sprays is a very exciting space. There's also some very innovative research happening now, looking at using visual cues, high-tech cameras and X-ray type photography, taking photos and scanning vision of plant leaves, where we can start to observe mineral nutrients, their distribution in leaves. It's going to be, the technology is still very early days, but when it gets, say, a decade or two away, when it's more also cheaper and commercialized, there's incredible opportunity to visually look at plants and get incredible detail of nutritional status. And what we're looking at here is, in this image, is this is a study on sunflowers and applying zinc. And this image over here, we're looking at the transect of the sunflower leaf, and we're looking at a time lag after an application of zinc, looking through the transect of that leaf, of the sunflower leaf. And here's your key here. You can see that the darker areas is low concentration of zinc, and we're moving to the lighter areas of high concentration of zinc. So over time, what you're seeing after a foliar spray, what you're seeing here at this 1, 3, and 6 hour mark, you can see the zinc concentration increasing, moving from blue to red. And we can see the zinc really is moving into those sunflower leaves. But you can see what's interesting with sunflowers, we're seeing these particularly hot spots, these white hot spots right here. This is the trichomes on the sunflower leaves. And so you can see that it is actually the trichomes which appear to be the most important site of absorption of that zinc. The zinc is primarily being absorbed through at the base of the trichome, where then it enters the leaf, and then you can see the leaf will turn red, where the zinc is then getting distributed throughout the rest of that leaf. So we're just using a special camera here to visually see this and identify where that zinc is. Over here on the right side, you can also see quite a neat visual. We're looking at comparing zinc sulfate versus a nano zinc fertilizer. So here we can see 3 hours versus 3 days after putting these droplets on the leaf. So you can see 3 hours after exposure, there's a bit of absorption of the zinc. But when we compare the 3 days here, we're comparing zinc sulfate versus nano zinc oxide. Here's the zinc sulfate, here's the zinc oxide. You can see that in this instance, that the zinc sulfate has been much more effective at being absorbing, getting that zinc into the plant versus the zinc oxide form over here, where you can see we've changed the color very insignificantly, a very small amount. We've moved from dark blue to light blue versus all the way up here. So, you know, this is a visual cue here, and this is a very exciting and emerging technology. So I think I just share this purely for interest's sake to highlight that there are more interesting tools emerging, and when we couple these kinds of techniques with the evidence base on the links of nutrition and disease, I think we will gain make a lot of ground and gain a lot of traction in this kind of precision nutrition approach, not just for managing nutrition for nutrition's sake, but for pest and disease management as well. So I wanted to say a few words, particularly on silicon. I'm using a silicon as a little quick case study with the discussion of nutrition. And just to say that silicon, although it's not an essential mineral for plant growth, it is very beneficial. And it is known to be a fantastic stress reliever in plant growth. It helps to relieve a whole host of stresses, both biotic, so insects, disease, fungus, bacteria, all sorts of invading pests. It's known to enhance, but also a lot of environmental stresses silicon can also help with, which is not the topic of today, but things like salinity, sodicity, heavy metals, heat stress, frost stress, droughts, waterlogging, you name it. Any environmental stress, silicon can enhance the plant tolerance of those stresses. But here we're going to talk about its role in pest and disease management. So as I mentioned, silicon has two modes of action. It's both primary and secondary that we touched on earlier. So it is cell wall reinforcement, linking in with calcium and boron, forming these structural compounds that get deposited in the cell walls to make those cell walls more robust. Silicon also accumulates underneath the cuticle. It's called a cuticle silicon double layer, where it will accumulate under the cuticle and also help to reinforce that. But then silicon also has a role internally. It can also induce or activate that immune response in the plant as well. So helping to signal the production of all of these defense chemicals within the plant. And this is just a visual to highlight. Here we have without silicon, the fungal spore can land on the leaf surface, and then the fungal hyphae will germinate and penetrate down through into those cells. Versus here, where we have that silicon layer that I mentioned deposited under the cuticle, this acts to reinforce, to thicken that skin of the plant. It makes it tougher, and therefore that developing fungal spore, that fungal hyphae cannot penetrate down through that layer. It will then be deflected away. thereby protecting the plant physically through just a physical barrier against that disease organism. From an insect pest, the same is true. This is from an interesting study here looking at some of these different silicon accumulations and which insects it helps against. So when we have an unprotected plant, we can see that the chewers can penetrate through, and our sap-sucking insects equally can penetrate through. When we have that surface deposition, the silicon layer under the cuticle that I mentioned, that does help to stop feeding insects, the chewing insects. That does help against those. However, it doesn't help against the sap feeders. They can still penetrate through that first layer and get through into the sap stream. We also see silicon accumulation within the cells, inside cells. And again, that helps against the chewing insects, but less against the sap feeders. Whereas once we move to that cell wall deposition, the silicon, the calcium, the boron that get deposited in the cell walls, that style of reinforcement of silicon is the one that does also then help against those sap-feeding insects, those phloem-feeding insects. So, different modes of action they're highlighting, that different insects have different susceptibilities or abilities to either overcome that or not. So, but anyway, just to highlight that there's some interesting work being done here with silicon, and then equally, as I mentioned, the internal immune response. There's a huge body, if people want to go and explore this, there's a huge body of evidence on the lengths of silicon and a whole host of diseases and insect pests. I would encourage you all to do some Googling on this one. There's lots of really good reading on this. This is kind of a really good kind of review summary of some of these effects on insects. You can see that across the board, silicon accumulation in plants, silicon-rich plants, has a big impact over the borers. They're the ones that are very susceptible to silicon-rich. They're not very good at overcoming that plant defense strategy. So you can see a big reduction in boring insects when we have silicon-rich plants. But equally, the chewers, and as I mentioned, the sap feeders they're the ones that have that ability to that overcome that we just showed. So you can see there on this graph here, they're a little less compromised, but still overall, all of these insect types here, you can see they all are reduced in the presence of silicon-rich plants. They're all less problematic in silicon-rich plants. So that's a good kind of review highlighting some of those different strategies. And I put this one in there just purely for interest's sake. This is a bit of an old one, just simply a hydroponic solution culture study looking at a host of different wheat varieties and looking at what their natural silicon accumulation is. And so they're all growing in the same solution culture, same concentration of silicon, yet you can see all of these various different varieties on the bottom have very different abilities to accumulate and take up that silicon. So there is a nuance around varietals. We know that certain varieties are better accumulators of silicon than others. So if we want to take a proactive approach about breeding for pest and disease resistance, there is also an opportunity here. Different varieties do have different abilities, and this is just a nice graph showing how variable different varieties are in the nature of that silicon accumulation. This one, I just want to say quickly about the nitrogen one. I'm not going to do a big discussion on nitrogen today. There's a lot more to say about nitrogen, but I'm just going to cut to the key point here, as I was alluding to earlier, which was just to say that the classic view of nitrogen management for plant health is that plants take up ammonium and nitrate, and therefore, a lot of our fertilizers are driven around ammonium and nitrate-based fertilizers. Urea, of course, is the other one. And really the point is just to say that although plants can and do take up these smaller ionic forms, plants can also take up amino acids directly from the soil, organic forms of nitrogen. They can also take up small proteins called peptides. They can take these up. So plants can absorb these organic forms of nitrogen. And the point is to say that really when the plant takes up nitrate or ammonium, it doesn't really want either of those. It wants organic forms. It takes nitrates and ammonium and or urea, it uses them as building blocks to build amino acids, to build proteins. That's really what the plant does. So this is the crux of what I was saying before, when we supply too much nitrogen in these ionic forms, in these more simplified forms, without the supporting nutrients that help to convert those building blocks into the organic forms, into the aminos, into the proteins, that's when too much nitrogen accumulates in the plant health, free nitrogen, not complex organic nitrogen, but free nitrogen. And that's what compromises plant health. Okay, so the example here, just to summarize this, these are the other supporting minerals we should be thinking about. So in order for the plant to convert nitrates into amino acid and onwards to proteins, the plant needs molybdenum, it needs sulfur, and it needs iron, particularly molybdenum in this instance. So if you have a limitation of any one of these three minerals, you won't be able to convert those nitrates into organic forms. Therefore, you will accumulate nitrates, and that will be attractive to insect pests, particularly sapsuckers in the example of nitrates. But similarly, here we have ammonia or ammonium. The plant requires manganese or magnesium to convert those. Or if we're talking about urea, the plant requires nickel to convert any of those building blocks into the organic forms. These minerals are essential catalysts in helping to convert that. So the point about nitrogen is just to say, don't manage nitrogen in isolation, manage it with these other supporting minerals in mind. That's the take-home message. So use your tissue, use your sap analysis, check for these other minerals and make sure they're in adequate supply so that you ensure you're converting your unfavorable forms of nitrogen into the more favorable forms, which enhance not only your protein and your quality from that point of view, but also your plant immunity. I'm not going to dwell on the refractometer. I'm sure you all probably have a pretty good understanding of it. I just want to link this discussion about nutrition and immunity to just say, yes, we can use a refractometer to basically measure photosynthesis, measure how well the plant is photosynthesizing. And we can look through the Brix meter and we see things like this. This would be a plant that has a nutrient limitation. It's got a very low Brix and a very crisp line that you can see here. So it's lacking the minerals that are the catalysts to help the plant synthesize those various sugars, carbohydrates, proteins, etc. And over here on the right-hand side, you can see we have a high Brix with a nice blurry line. So many of you would be familiar with this concept. I just want to then bring it to this slide that we showed earlier to just kind of link this discussion, is that when you see plants that have that Brix reading with a very crisp line, back here, that very crisp versus blurry, there the crisp line is indicating more simpler sugars, smaller stuff, not the complex stuff. This is what we're really chasing. It's more of the glucose, that building block molecule. This is your link with that crisp line. So you've got sugars there, yes, but they're smaller, simpler. They're not linking together to then form the more complex, real quality and immune enhancing stuff. So when you then see the blurry line, as indicated there, that's the blurry line. You're really then picking up on more of these complex and diverse products of photosynthesis. Okay, and again, the point is to say that if you don't have adequate mineral supply, you can't get these here. Okay, so your minerals are the catalysts. With the Brix meter, you are indirectly measuring plant photosynthetic capacity. You're measuring plant products of photosynthesis, and therefore you're indirectly measuring the plant mineral status through that, because you can't get these without minerals. Okay, so the refractometer is, and that's a big discussion. We're not going to talk about refractometers and the nuance of using the meter. There's a lot of nuance to using it in field, sunny days, cloudy days, dry, wet, etc. I'm not going to go into that, but just to highlight that it can be a useful tool when used properly to help tap into this discussion around not just photosynthetic capacity, but also plant health, plant immunity, because that blurry line is where you're also measuring these protective compounds, these defense chemicals that the plant is also producing. And so if we then think about it in the context of insect or disease resistance, you know, the principle is the same. You know, we need nutrients to photosynthesize for plant growth. We need also these minerals to synthesize these insect repellent compounds, the anti-feedant, anti-herbivory, cell-strengthening, deterrent chemicals, the volatiles, all of these protective defense chemicals. Okay, so from an insect resistance point of view, it's the same. We've got to be optimizing nutrition, which optimizes photosynthesis, which also optimizes the production of these immune-enhancing compounds that protect the plant against insects. And it's the same when we think about disease. Again, it still comes back to mineral catalysts, macro and micro mineral catalysts, which then help the plant to produce, again, from that building block, that glucose building block, it produces these other more disease-enhancing compounds, the antibiotics, antimicrobials, again, physical barriers, cell strengtheners, defense chemicals, et cetera, et cetera. Okay, so these are the real compounds you're chasing that actually give true immunity, and these are the ones that give you that blurry line on the Brix meter, but you can't get any of those without adequate supply of the essential minerals for plant growth. Okay, so that's kind of our nutrition half. Let's segue into a little bit of discussion around biology. And this is then just to say that there are a whole host of biological interactions when pathogens or other beneficial microbes, when they interact with plants, there's a whole host of signaling and communication that happens around the plant. And here's an example here, where just using diseases as an example, it could also be true of insects. When a pathogen lands on the leaf surface and begins to cause disease, if we have a soil-borne pathogen beginning to cause disease on that root system, that triggers the plant detects that, and it triggers a systemic communication response throughout the plant to turn on its immune system and protect itself against that invading attack. And what that systemic response is typically means that plants will detect that. They will usually then change their root exudation pattern profile, and they will begin to recruit beneficial microbes from the soil. Many of these other beneficials that are also in the soil that then are activated by those root exudates, by those stress calls. It's kind of a stress alarm call that the plant will release, and they will respond. The beneficials will respond. Those beneficials will produce a whole range of metabolites, chemicals themselves. They will exude all sorts of protective compounds into the rhizosphere, and these chemicals can sometimes be directly absorbed and taken up and also helped then sent to the site of infection to protect the plant, or these are also inducing signaling molecules themselves that help to turn on other biochemical cascades that again trigger the response, trigger the production of these protective biochemicals. So we're going to kind of move and think about some of these kind of processes as we link some of the discussion around the role of biologicals and biology in pest and disease prevention. So here's some examples of how biology can help with disease, a few modes of action, if you will, in terms of how they protect. So one is a very simple competition, competitive exclusion. This is where beneficial microbes will compete against pathogens for space, for food, for nutrients, a very simple competition process. But many of the beneficial microbes can also directly antagonize pathogens. So they can produce a range of these microbial exudates, microbial metabolites, these biochemicals that microbes release that can have antibiotic, antifungal type properties, so suppress pathogens, various kind of inhibitory compounds. Some beneficial microbes even prey on disease-causing microbes. So, for example, trichoderma would be a good one, a predatory fungus, a fungus that eats other fungus, digests other disease-causing fungi for its growth. So some actually eat each other in the dog-eat-dog world of the soil microbiome, soil food web. And then lastly, these, as I just touched on earlier, many of the microbes can help to induce a plant response, a plant immune response. So they're indirectly helping the plant fuel that immune system and then helping the plant combat and ward off the invading attacker. So in terms of managing biology, practically managing biology, we have kind of two schools of thought that emerge here. One says, let's stimulate or let's feed, let's nurture, let's look after the native organisms, the organisms that are always present, already present. So here we're saying, well, let's optimize soil health, let's feed the soil, let's create plant diversity to encourage good soil, diverse microbial populations. So here we're kind of implementing strategies to improve soil health, to stimulate, to feed, to nurture the native organisms that are already there. Then we have another school of thought that then manages through inoculation, says, well, let's introduce new populations of microorganisms for specific function, for a specific goal. And these could be soil applied or plant applied, foliar, seed treatment, or applied down into the soil. Now, there's a big debate on this. I don't know if there's necessarily a right or wrong. I think there's a lot of context dependency on this in different scenarios. My personal opinion on this is I think a combination of both is a good strategy, but I would say that I think our first and foremost strategy should be the top one. We should focus primarily on feeding who's already there, the native organisms. They're obviously adapt to being there, so let's just nurture them. I think that is the preferred and primary strategy. But there are, I believe there's also many instances where an inoculation can be warranted, perhaps in the short term, maybe to kickstart the process, maybe a short-term thing, not a long-term thing, or in certain scenarios, if we're requiring a specific function. If you're looking for lots of nitrogen fixation, well, use a nitrogen fixer. If you're looking for disease control, well, then apply a predatory fungus. For very specific targeted functions, I think some of these inoculants can be useful. But again, I know there's debate on this, and again, yes, I agree. We should exercise some caution about introducing foreign organisms into new environments where they may become, alien species into that ecosystem where they can potentially become damaging as well. So I acknowledge, be cautious, but I guess just think of rhizobia, look at that as an inoculant. It's been extremely successful. One little bacteria has had a massive, vast implication on agriculture globally, commonly used as an inoculant, and often used in the short term until they build up in the level of the soil that they're no longer required. So they're one example of a very successful one. So if we were to then focus on the feeding microbes approach, things like molasses, sugars, carbohydrates, fish hydrolysates, these are all good food sources to feed microbes. Seaweed, kelp extracts, humics, fulvics, various plant extracts, or as I said before, designing the production system with a little more plant diversity. All of these things help to encourage, feed, support, nurture the native organisms. If we were to introduce new populations, again, as I was touching on there, you've got things like commercial inoculants or DIY inoculants. So commercial inoculants, you might be buying in things like mycorrhizal fungi, again, N-fixers, P-solubilizers, effective microbes, all these kinds of things, whereas you can also make your own. And I think these DIY inoculants perhaps offer a nice middle ground in the debate between feed natives versus introduce new. At least with a DIY inoculant, you're manufacturing it on your own property, in your own ecosystem. Therefore, you're likely to be recruiting and colonizing the local native organisms and then just culturing them up. So I think they have, perhaps also a good middle ground and a useful role. So here we're talking about potentially mycorrhizal fungi, but that's pretty labor intensive to manufacture them your own. But other things like indigenous microbes or compost production and compost extracts, these kinds of things, I think they offer a good middle ground in terms of trying to introduce more diversity or targeted use of microbes as inoculants, but manufactured on your own property. So that's then to say that there are many plant ecosystems that microbes live on a whole host of different plant surfaces. And this is an emerging area of science and research at the moment, understanding that different microbes associate with different parts of the plant. And this is quite an untapped resource that will also help us use biology for pest and disease management in the future. So the two that people would be familiar with is things like the rhizosphere. I'm sure many have heard of the rhizosphere, the ecosystem around that millimeter or so, around the root surfaces in which microbes live and colonize and support plant growth. So the rhizosphere is, and the rhizoplane is the microbes that live right on the leaf surfaces. So this is a big one which we often hear about. This is a unique community of microbes that live around the root system. But there are many other microbial communities that we're only just beginning to understand and beginning to tap into that may well be very important for also this discussion on plant health. So things like the anthosphere, unique microbes that live on the flowers of plants. The carposphere, microbes that live on the fruits or the seed or the grain of plants. The colosphere, these are unique microbes that live on the stems of the plants. the Phytosphere, people might have also heard of, microbes that live on the leaf surfaces, and the Endosphere, microbes that live inside the plant tissues, synergistically, mutualistically within the plant tissues in which they live. And also then lastly, the Spermosphere. This is the seed microbiome, the microbes that live in or on and live around the seed. So all of these are quite unique microbial communities that we're only just beginning to understand, and they may also hold very important keys for us in helping us to understand plant immunity. And it's amazing, there's a lot of differences in each of these communities. You might assume that all of those microbial species that live above ground, say on the flowers, fruits, stems, and leaves, you might think they're all more similar to each other than those that live in the soil. Obviously, two very different environments, one above and below ground. An interesting study has shown that actually each of those microbial communities, for example, the microbes that live on the flowers, they have more in common with the soil microbiome than they do with those that live on the stems or in the endosphere or on the fruits. So equally, the ones on the fruits or the ones on the stems, they have more in common with the soil microbiome than they do each other. So they're really quite unique, and we're just beginning to untap this whole kind of space. And so just to say a little bit on seed microbiome, because this is an opportunity to introduce our new inoculants. When plants reproduce, when they grow, produce, and go through their life cycle, and then ultimately produce seed for the next generation, they actually have, during the growing season, they have these unique microbes that live on the leaf or in the endosphere or on the flowers, on the fruits, as I just mentioned. It turns out that the plant intentionally sends certain species of these microbes into the seed, and the seed is designed to carry those microbes into the new environment, wherever that seed will be dispersed through wind, through water. So when that seed then lands, it actually is a seed, it is a microbial inoculant in itself, and it contains some of these various species from the plant phytospheres, from those various ecosystems on the plant. Then those various microbes begin to interact with the soil microbes, the ones represented by black here. Then they begin to interact, and then that seed begins to germinate, and then that plant will grow. And those microbes typically will then migrate up into the above ground again, and the ones that were found in the soil will stay down in the soil. So this is just to highlight that in the debate and discussion about introducing new microbes, new populations of microbes into new environments, and the concerns around introducing alien species and these kinds of things, not to belittle that concern, it is an important one, but just to highlight that in a natural ecosystem, plants already distribute microbes with their seed. Plants are already spreading microbes into new environments as part of their survival mechanism. So when we as humans have already been trading seeds for many hundreds or thousands of years, the horse has already bolted. You know, when we traded seeds, we've already taken microbes all around the world already. You know, it's already happened. So just to highlight, it is a natural thing for that to happen, but sure, exercise some caution too, but equally, it is a natural process. So there's a lot of interest then in using microbial inoculants as seed treatments, and with good reason. These microbes here at the very beginning are very important. This is something called a core microbiome, these kind of select species that get carried through into the seed. And lots of studies are showing that they're actually critically important in a whole host of plant functions throughout the rest of the production cycle. So there's a lot of work looking at seed treatments to introduce some of these inoculants. And one of the kind of common ones I see many farmers using around are the use of things like liquid compost extracts or teas or slurries, you know, bringing a very diverse microbial inoculant and using a seed treatment there with that compost extract. Again, as I highlighted, if we're using a compost produced on farm, you're then culturing more native local organisms and then targeting that around the seed or injecting that into the furrow, providing that little inoculant there to grow, to when that seed germinates, those microbes are there and ready to grow. and grow with. Now, and again, just to highlight that when you're using a targeted strategy like that, you know, again, the classic argument against using inoculant says, well, why would you bother putting some microbes into the soil? It's a drop in the ocean. You know, you're putting a fraction of the number of microbes in that soil compared to the billions and billions that are already there. And people discredit the technique of that. And that's valid in the whole soil context. But when you zoom in to think about targeting on the seed or in that furrow, we're not trying to change the whole soil microbiome. It is a drop in the ocean, but when you target that drop right on that seed, well, you only need a tiny inoculant. You only need a few microbes there that when that root emerges and begins to excrete those root exudates, it will recruit those microbes. They will simply grow with that root. They're not going to grow into the soil. They may not change the soil whatsoever, but they will be there in that rhizosphere growing with that root. So you're not trying to change the whole soil. You're targeting and changing the rhizosphere. And the rhizosphere and the bulk soil are two very different worlds. So, you know, there's some interesting work happening with compost extracts, specifically as seed treatments or injection. And if we look at the literature on this, indeed, there's good evidence that shows that compost teas and extracts, they have been shown to have a whole host of suppressive effects on disease against all of the major diseases: Pythium, Rhizoctonia, Verticillium, Fusarium, Sclerotinia, Phytophthora. They've been shown to have some suppressive capacity against all of those. However, it's not to say that they can always control those diseases or eliminate those diseases, but they have suppressive effects. And however, those suppressive effects are variable. And that's the nature of a compost product. It's not the same inoculant time after time. It's a naturally occurring product. It is variable. When you make an extract, the concentration of microbes is variable. When you brew a tea, it's variable. So you cannot guarantee that it will behave the same every time. And again, that's the argument people will use to not use the strategy. However, it has suppressive capacity. And as one tool in an integrated approach, then I think it has legs. If you're relying on it for sole disease protection, maybe not. As a piece of your integrated strategy, maybe yes, because it does have some suppressive effects, and you can use that with other strategies as well. So I'm going to skip over these few slides because I can see I'm getting a little tight on for time. This was a study just to say that we know that it is the living component of the compost that brings the protective effect. It's the biotic. We're comparing sterilized and unsterilized compost, and showed that the unsterilized, the living compost, is what had the suppressive effects in this example. But this is another study that looked at vermicompost extracts. And in this study, they drenched the soil with vermicompost extracts and didn't apply any to the foliage. And here they found that when they drenched the soil with vermicompost extracts, it induced a suppressive effects on aphids, mealybugs, and mites. So these pests were less attracted to the host plants, and it even killed off some of them. And so here the authors suggested that it was not a living factor. In fact, it was a non-living factor, an abiotic factor. And here they put it down to these compounds called phenolic compounds that were extracted out of the vermicompost, that the plants absorbed these phenolic chemicals, which were then translated and which then protected that plant, translocated and protected that plant against pests. So I share these two examples to say that it is both a biological protection, but there is also a chemical protection as well through some of these other biochemicals that come out of compost extracts that can also help protect the plants. So it is indeed both effects. Mycorrhizal fungi would be another one that have been well shown to protect plants from disease. Very simply, when mycorrhizal fungi colonize that root system, they, of course, produce all of these fungal hyphae, and that simply outcompetes space, takes up space, helps to outcompete pathogens who are trying to get to that root system to ultimately, or nematodes, to ultimately cause disease. So mycorrhizal fungi also help the plant to mount its own defenses. So mycorrhizal fungi can induce that immune response we talked about and help the plant from disease as well. Mycorrhizal fungi can also help to plants in a population to communicate to each other and distribute stress signals or alarm signals. So this was a really interesting study on tomatoes, where they grew the tomatoes in shared soil, but they put these kind of bubbles over them. So the tomatoes were not sharing airspace, but they were growing in the same soil. And in these two tomatoes, on one of them, they intentionally inoculated the tomato with blight. And of course, that plant stressed, it produced an immune response. It would have released a whole host of volatile compounds into the air, but they were contained within the bubble. So there was no air communication from plant to plant. They showed that actually those stress signals were sent down to the roots, given to the mycorrhizal fungi, and the fungi then transported that stress signal over to this other plant, which was not challenged by the pathogen, which was not sharing airspace with the challenged plant. And yet they measured an immune response in this plant where 6 genes known to code for these defense chemicals were activated. So mycorrhizal fungi also play a role in sharing that immune response. So then in terms of triggering this immune response, managing the plant, there are many things in which strategies we can use to turn on plant immune system: biofertilizers, biostimulants, various botanical kind of plant extracts, natural pesticides. And all these things ultimately help to unlock this phytobiome, this plant microbial world, in which helps to then induce these kind of immune responses. And it turns out that an immune response primarily really happens in the soil. So this was an interesting study that challenged the plant with an insect pest, and then they were measuring changes in genetic expression in the leaf versus in the roots. And you can see that overall, even though the insect attack happened on the leaf, here's the amount of kind of changes we saw in genetic expression in the leaf versus the change in genetic expression in the root cells. You can see that there was a much greater impact or change in that genetic expression down in the root system versus up in the leaf. So plant immunity really exists in the roots and in the soil. It is the plant recruiting the microbes in the soil through those stress signals and root exudates that help to recruit and induce beneficial microbes to help the plant then mount those defenses, as I touched on earlier. So it really is all a rooting response. So how can we help to support and induce this resistance in the plant? Well, as we've touched on, nutrition. So silicon, I mentioned, manganese, copper and boron. Those 4 in particular, very important for priming those internal bioprotective chemicals. But as I mentioned earlier, also really a deficiency of any one of the essential macro or micro minerals, which will then compromise various plant metabolic pathways, ultimately will lead indirectly to a reduction in plant immunity. So any deficiency is the real answer, but if we want to target some of the ones we discussed, particularly silicon, manganese would be high up that list. Biostimulants, kelp, the cytokinins have been shown to trigger an immune response. Chitosan, a compound that's extracted from shells, insect shells or crustacean shells, sea organisms, these kinds of things. So any of your fish hydrolysates that have crab or shell material or the insect frass, insect waste, any of these kinds of things are rich in chitosan. This compound has been shown to induce a very strong immune and protective response within the plant. So this can also be foliar sprayed over the plant for inducing that effect. And then a whole host of plant extracts. I mean, you name it, essential oils, I mean, garlic extracts, rosemary, I mean, various different plants, you know, the classical nettle teas, you know, these kinds of things. I mean, there's a ton of plant extracts that have also been shown to have various immune enhancing responses. And then some of those biofertilizers, compost extracts I mentioned, a great tool to provide a diverse inoculant. But if we want to target down to something a little more specific and target those disease antagonists, specific disease antagonists, we could use things like Bacillus subtilis is a well-known bacteria that's a great disease fighter. Pseudomonas species, Trichoderma is the one I mentioned, the predatory fungi or mycorrhizal fungi. Those are some of the big players that are particular disease antagonists that we know about. A few very quick words on diversity. This is just to highlight that one of the central reasons that we have problems with pests and disease is a fundamental design flaw of how we produce our crops, which is the monoculture system. when you have a monoculture system, you have not just the same species, but usually the same variety of that same species, so extreme double monoculture. If that variety is susceptible to a disease or an insect, well, of course, it's just too easy, particularly for that disease, to spread through that crop. It's the monoculture that just, the uniform susceptibility that will enable the rapid spread of that pest. It is the design flaw of the monoculture. As soon as we move to any slight more diverse system, well, very simply, we have diluted out the host plant with other plants, and therefore it's more difficult for the pest to then spread like wildfire because there are more non-hosts in the system. Okay, so that simple step away from a monoculture can have big impacts on pest and disease resistance. I could talk about this for a lot more, and this is not the topic of today, so I just want to share quick examples to put it in the mix to link to our closing point. But this was a review looking at disease in intercroppings, highlighting when comparing monocrops versus intercrops, showing that intercropping reduced disease in 73% of the incidences. Out of 200 studies that they looked at, 73% of the time, intercropping led to lower disease resistance. Okay, so just jumping from 1 to 2, one small step to an intercrop can begin to lower disease pressure. Not in 100% of the cases, but in this study, as it says, 70-odd% of the time, that will lead to a favorable reduction. Nematodes were the one that was the opposite. They say intercropping can enhance nematodes, according to the study, but with your diseases, broadly, it leads to a reduction. And this is also another interesting one, looking at, as we increase plant species diversity, plant host species diversity, we also then, as you would assume, we see an increase in pathogen diversity. So as we have more hosts present, of course, more host plants mean more plants that would encourage more pathogens who attack that host into the mix. So we're seeing increased number of plant hosts, therefore increased prevalence of those disease organisms that attack that plant. However, although you see an increased occurrence of the disease, presence of that organism, the overall infection of the plant host actually declines as we increase plant species diversity. So, it's this, the key point to say is just because you have a pathogen present, it doesn't mean that it's causing a problem, that it's causing disease. And this was a really interesting study that highlights that point. Don't be alarmed just because the pathogen is present. Like you can see here, the overall infection rate, the overall effects on plant growth or disease plant susceptibility declines as we increase plant species diversity. And this is one specific example, just to show you intercropping faba bean with wheat, just as one small example. So here we're looking at a meta-analysis, so summarizing a whole host of other studies to quantify the disease suppressive effect of intercropping cereals with legumes. Okay, they were also looking at different rates of N fertilizer. They showed that intercropping reduced the disease incidence by 45% on average. So that step from 1 to 2, just simply adding the bean into the wheat or the wheat into the bean, lowered the disease pressure on average by 45%. So they saw reductions in yellow rust and mildew in the wheat and chocolate spot and wilt for the bean. The results show that intercropping has a substantial and consistent effect on disease incidence in cereal faba mixes across those studies, but it is not sufficient to provide complete control. Intercropping is therefore best used as a component in an integrated approach for managing plant disease. And I think that's the crux of the point. I wouldn't pass up a 50-odd% reduction in disease pressure. I think that's an excellent result, particularly if I was combining that with, say, again, a compost extract that also will not give complete control, but may give another 20-odd% control. That's going to be another piece of the puzzle. Then we're going to manage nutrition with some manganese and some silicon. That's going to give us another percentage of control. And that's the point of an integrated pest management approach, is that you're bringing all of these together for that desired goal. So ultimately, we have to come back to asking ourselves, about treating symptoms or addressing root causes. And this is just a thought exercise, and I'm being, you know, intentionally pushing your button to kind of just ask questions in this thought exercise here to question how we do things, question the fundamental kind of status quo on pests and disease. But, you know, just thinking about it in this context, is the disease caused by, if we're looking at root causes, is it caused by poor plant immunity, poor plant health, a lack of antagonistic microbes or beneficial microbes? Is it caused by an absence of fungicide? You know, and ask yourself that question: what is the root cause? If I apply this fungicide, does it address the root cause? Will the disease not come back? And of course, the answer is no. You're treating a symptom with that chemical. You're not addressing the root cause. Is my insect caused by poor plant nutrition, poor plant health, lack of beneficial insects, predators? Is my insects caused by a deficiency of insecticide? And of course, the answer is no. And if it's not caused by a deficiency of pesticide, then we have to push ourselves to the next question. Well, what is the root cause? And that then opens this discussion into these other ideas of plant health, looking at nutrition, looking at biology as other pieces of that puzzle. But I acknowledge, if you're seeing symptoms, you have to treat them too. It's not just about addressing root cause. That's your long-term game. You still have to treat symptoms. If you're seeing symptoms, you've already lost productivity. But you have to stop the spread also for next season's crop. So, you know, there are softer alternatives, the substitution, using nutrition, as I mentioned, using other biologicals. These can be important transitional strategies. I mentioned some of those botanicals or organic or natural pesticides. If you're using any of these, synthetic or natural, a biocide is a biocide, you know, irrespective if it's natural or synthetic. So if you're using any kind of a chemical, a sterilant, you are compromising some of the beneficials as well. So you may need to therefore re-inoculate the leaf or the soil to repopulate the beneficials. And that's that argument about, do I feed natives or support or use new inoculants? And I think in certain examples, if we've sterilized the leaf with a fungicide, well, maybe there is an argument that we need to reapply some beneficials to help repopulate that. So, of course, prevention is always better than cure. So let's focus on those longer-term strategies, healthy soil, however we define that. And that's another big debate too. But, okay, think about your mineral supply, your mineral balance. Are those nutrients available to the crop? Those nutrients, hopefully you can still hear me. My Wi-Fi's playing up, I can see. Optimize your nutritional status of the soil. Think about soil structure, soil aeration, soil infiltration. Think about biological activity, diversity. Think about organic matter. Improving all of those is going to improve soil health. That's going to improve your disease resistance, pest and disease resistance in the crop. And also think about a healthy plant. Optimize its nutritional status, do tissue analysis, make sure it's got the minerals, protect that plant with beneficial microbes. Okay, in summary, I apologize, I've gone a little over time here. Hopefully we've got time for questions at the end now. In summary, I would say integrate multiple strategies for pest and disease. There is no one silver bullet. So use nutrition and biology, use plant species diversity, use all of these things as integrated approach. Minerals function to support those immune systems of the plant. So do a leaf test, sap test, do in-field monitoring, refractometers, use foliars, use seed treatments, address nutrition, address biology, silicon, calcium, boron, manganese, copper, nitrogen. We discussed all of those. If we think about biology, use feed your natives, plant species diversity, seaweeds, fish, molasses, these kinds of things. Inoculate where appropriate. Optimize microbial diversity with compost type extracts, maybe using those inoculants as specific antagonists. And again, of course, overall improving that soil health, using soil health principles. These kinds of things are going to help improve both the nutritional fertility and also the biological fertility and the physical fertility of your soil, and therefore improving soil health overall. Okay.
Excellent. Thanks for that, Joel.
Sorry, I went a bit over time.
No, that's all right. We'll hook straight into some questions. I really like how you ended the ended the presentation with we really need to start looking at the cause of these issues, not just treating the symptom all the time and through going that way. But anyway, there was a question emailed through prior to the webinar that on the Russian wheat aphid. So Joel, if you wanted to just quickly touch on that, the one that we emailed through prior to the
Yep. Sure thing. Yeah, it's quite a lengthy question, so I won't really read it out. But it was, yep, as you mentioned, susceptibility to the Russian wheat aphid, and I can't say that I have the specific answer to the specific problem in this context. But as you've kind of just heard in the webinar there, and as we've just touched on, I would be taking that holistic approach. I would be using multiple strategies, but we're looking at nutrition and biology. But if you're talking aphids, there's some good examples out there linking the presence of aphids, particularly to plants that have high nitrates in them. So, I'm not saying that is the case in this instance, but it could be a likely candidate that you have a nitrogen imbalance in the plant. So too much nitrates. Sap-sucking insects are particularly attracted to the nitrates. So if you've got an excess of nitrates in the plant, that is going to suggest you may have a limitation of molybdenum, most likely, or sulfur or iron. Those are the other three minerals that help to convert those nitrates into ultimately on to nitrite, then ammonium, and the amino acid and protein. So I would suggest looking at your nitrates. I would consider a foliar spray of molybdenum, possibly sulfur and or iron. As I touched on in the presentation, we also talked about the links with silicon, and I showed that the sapsuckers can bypass some of those silicon protection in the plant, but still there is that evidence that shows that when that silicon is deposited right in those cell walls, that does also stop the aphid. So, and those sapsuckers. So I would also maybe perhaps consider looking at silicon as well. And you could do foliar sprays of things like potassium silicate, or silicic acid is another input that can also be applied. So yeah, I would be looking at nutritional things like that as my first guess, but ultimately I would do some kind of a tissue test or a sap analysis. I would look at the mineral status. I would address whatever is actually limiting according to the test. That's what I would do through a foliar spray for rapid response. Yeah, that's what I would do. And hopefully you've picked up a few other ideas from watching the presentation.
Yeah, a few years ago, we actually had a fairly big problem around our area in rice with the Russian wheat aphid, and we sprayed out on the crop. It was TM Agriculture and a liquid vermicast product, and that seemed to get rid of them pretty much overnight. And the people who used an insecticide, they quite often got a reinfestation, whereas ours, with what we did, that didn't happen. So that was really interesting. So, yeah.
Yeah. A vermicompost extract is going to help prime some of that, more of that immune response through triggering an induced resistance. Many beneficials can induce that resistance in the plant and trigger that change in root exudate profile. So yeah, something like an extract, although it may not directly antagonize the aphid, indirectly it's going to help induce that immune response to help that plant protect itself.
Yep. Excellent. We better keep rolling on. Got one from Graham Hendy. Question, basically, I think we covered a fair bit of it. Yeah, but what can we use on the seed instead of fungicides and insecticides, he was asking. So we tend to use a biostimulant or something like that.
Yeah, that's right. And I touched on, I approached that to think of three things. It's minerals, microbes, and carbon or biostimulant. Yeah. And in the context of a seed treatment, I lean more towards the biostimulant and the microbial part, and a little bit of nutrition as a supporting thing. So yeah, in that instance, I would focus on things like, seaweeds, extracts, humic acids, these kinds of things, molasses, but compost extracts. That seems to be what most farmers are using, a mixture of general biostimulant, whatever, molasses, fish, humic, whatever you've got kind of handy. and a compost extract of some kind, or a brew microbial consortium if it's a commercial product. And yeah, mix those together. Maybe a little bit of manganese is really important for germination, a little bit of nutrition in that sense. But yeah, I would lean towards some kind of a biostimulant, bit of seaweed, and some microbial. Yeah.
So Kieran, have you got anything to add to that? I know you did a bit of a thing, biostimulants versus pickle there a couple of years ago, and what was?
Yep. Oh, absolutely. Well, the biostimulants, we've just found we get a far greater and more robust germination immediately. So actually, Darren's just brought up on the screen. So this is one of my wheat crops. I live in the northwest of New South Wales, and this is a paddock that actually it's been farmed since the 1970s. So it's fairly tired, worn out country. We've been using a biostimulant on it for several years. But a good example of, I do a lot with Brix and Refractometer measurements and just showing that. And I noticed there's another question coming up from Anne about rust in wheat. So to answer that, and in regards to how do you manage rust in wheat crops, I could notice there was some rust starting to creep into this crop. And it was also just looking a little bit hungry. It was a bit yellow and a bit stripy. So I decided to apply a foliar fertiliser to it. So it consisted of NPK, magnesium, sulphur, iron, boron, zinc, molybdenum and manganese. And this was applied with fulvic acid and seaweed. So they're a product that work together to really help as a tonic for that plant, but also to increase the efficacy of those nutrients being delivered into the cell walls. And I did notice, so the Brix readings before we applied the foliar to this crop when it was looking a little bit sick and had some disease on the leaves was 8. And 6 days after the application of the foliar, I did another Brix, which you can see here, and it had gone up to 20. And you can see it's a little bit of a blurry line as well, which is a nice indicator as well that there's some good nutrient balance there and combination of all those macro and micronutrients. So that really does help. It just keeps, we've moved right away from using pickles on our farm, and a lot of the farmers who I work with no longer use pickles. They don't have any issues with disease. If they do, we always try and work in first by using a biostimulant or a foliar before we go in with the big guns in regards to a fungicide, because as Joel will back up, they can be a lot more detrimental and interfere with photosynthetic activity and general plant health. So that's the way that I approach it.
Yeah. So Darren, can we go over time, or do we get cut off at?
I think we can keep going. We've got 7 minutes, mate. So just whip through a few of the questions, and if for some reason that does happen, we will sort of answer each question individually. But yeah, let's just keep going.
Carol Lim sent through a question: can I use kefir to introduce probiotics into my soil? So I know we've used kefir to make EM because it's got those specific bacillus bacteria in it. So I suppose it can, but yeah, what's your thoughts on that, Joel?
Yeah, lactobacillus. Yeah, you can use it, and other yogurt and milk, kind of dairy products and things. Yeah, I've heard of some good results with using kind of dairy products based sprays and mixes, offering some good protection against things like anthracnose in a horticultural context. But yeah, so yes, you can, and yeah, that's, you know, there's a whole host of those kinds of fermented style products, biofertilizers, and a lot of the bioferments that are coming out of Asia. A lot of those kind of strategies. Yeah, I think there's a lot of opportunity there to use those kinds of things. And so yeah, you could use kefir, especially if it was kind of homemade. I think generally we see that some of the probiotic products like yogurt and things on the supermarket shelf, they do contain bugs, but often not super high concentrations. So I think if you were to make your own as well, it would be even better. But yes, some of those microbes are also beneficial in a plant and soil context.
Yep, no worries. So Margot sent through a question. The plant cuticle is a protective layer. So what effect do surfactants have on the health of the plant and like when you're using foliar fertilizers and that sort of things? Do they damage it or not?
Yeah, so they can do. It's a mix to that. So yeah, you're right. Some of them can indeed kind of erode or dissolve the kind of the cuticle and actually lead to greater pest and disease susceptibility. It's not to say that all of them do, but yeah, indeed, that is true. Some of them can do that. So yeah, I guess you could exercise some caution with those. I think those kinds of things, stickers, they can be very important if there's some bad weather, that kind of thing coming. But I think generally I would lean towards, fulvic acid as a really important chelator, which then helps, performs a similar function, but, kind of helps to improve the uptake and get your nutrients into the leaf more effectively, more rapidly. So I would prioritize, things like that, seaweeds, molasses as chelating agents, and if there's some inclement weather, then that's good, because that's pretty much what we do. So Santosh, he sent through these silica protected plants. Are they the produce, is that safe for animal and human consumption? Yeah, it is indeed. Silica is a very important nutrient for human health, one that often is not talked about, but has a lot of links with skin and nails and hair health. Silicon accumulates in these parts of our body, and also particularly in your gut lining, very important in influencing absorption of nutrients through the guts as well. So plants already contain silicon. It's already there in them. It's a very normal process, a normal thing for them to contain. All we're really doing is elevating those levels, but not to any excess that's going to induce any negative consequence on human health. No, it would be a beneficial thing.
We touched on this, Kieran touched on this one before. There's been a huge issue with rust this year, especially down here in sort of southern New South Wales and Victoria. So what steps can they take next year to alleviate that and maybe not use as many fungicides?
Yeah, I think there's, a bit of info out there highlighting a bit of a link with potassium and rusts. Not to say that it's guaranteed to be it in this example, but there is some evidence that shows that potassium rusts are more prevalent in the potassium deficient conditions. So that might be something I would look out for, but really more specifically what I would do is be proactive about leaf sampling, doing your tissue or sap analysis. I would do it early on as a proactive approach, and I would address whatever limitation comes up. That's what I would be doing, addressing the nutrients that are actually required. But yeah, potassium could be a culprit. I've seen a bit of evidence on that. And again, silicon is a master mineral that will usually help as well. So again, things like potassium silicate is a good example of input that would supply both of those.
Yeah, no worries. And for people who are a bit unsure, like I know I answered this question a bit, tissue testing is not a difficult thing. So if anybody wants to know anything about that, just send an email to Darren or get in contact with myself. We can organise a tissue test for anybody quite simply. It's not difficult to do. It's pretty easy, and it is a really good tool to use. So Bryce has sent through a question. Does your sap pH trending towards neutral assist in pest and disease resistance?
Mm-hmm. Yeah, sure. So that's a common technique that a lot of farmers use to optimize the sap pH around this kind of 6.4. The idea came out from Bruce Tainio, and I know many people use the technique. Ramsay promotes it a lot, obviously, and it's an interesting one. Bruce mentioned he kind of developed that whole thing from observing weeds. And weeds typically often have good plant health. They grow with good vigor. They don't suffer a lot of pest and disease problems very often. And Bruce kind of tuned into this sap pH thing from measuring weeds. I've done quite a lot of measuring of weeds myself. of the sap pH. And yeah, for me, they're always above 6. I've rarely found a weed that's below 6. It's always 6.1, 2, 3, 4 in that kind of range. So yeah, you know, I think that my crude answer is yes. I think there's definitely something to it. You know, I think there's sometimes other greater environmental factors that might override, or, you know, some of those kind of variables. But yeah, I know a few farmers that really use the approach and have said it's been successful for them. So managing the nutrition to try and get that pH in that zone for plant health. So yeah, I think there's something to that. There's not, from a strict kind of scientific literature evidence on the literature, you know, there's very little. It's more of an anecdotal thing that many people use, but many farmers seem happy with and feel that it's helped them just fine tune the, you know, nutrition is a big topic, and it just helps to kind of fine tune that into a focus. So yeah, I think it's worth exploring. Yeah, worth using.
Yep. And there's one more. This probably is not so much on the pest and disease, but what a question's just come through: capeweed and those sorts of things. What's the nutrition in the soil like? What are they getting out of the soil? Why have we got these weed burdens? That's a whole new topic, but
next time, yeah.
Yeah.
Yeah, you know, so there's one school of thought that looks at that from a nutritional point of view and says that certain weeds will accumulate to and indicate certain nutritional imbalances or deficiencies, that kind of thing. So I don't know specifically on dock and cakeweed what maybe those nutritional links might be. So I don't know, but some would say that there would be some kind of a nutritional link there, and helping to address that or overcome that would help to lower their vigor, that weed vigor. The biological view looks at that more so from a fungal to bacterial kind of point of view, and says that a lot of weeds are generally more earlier successional plants, not all of them, but generally more earlier successional plants which thrive in more bacterial dominated soils. So as we shift that fungal to bacterial balance to a more kind of 1-to-1 or slight fungal dominance, that then selects for later successional plants, which then can outcompete the earlier successional plants which thrive under higher bacterial conditions. So those kinds of things, therefore, could, from a biological point of view, could indicate a microbial imbalance and therefore most likely a fungal deficiency. So trying to increase more fungal content of the soil. You could do that with fungal inoculants, fungal compost, fungal seed treatments, compost applications. You could do that with increasing plant species diversity. That's been shown to enhance fungal prevalence in the soil. So some of those kinds of things I would then try. And then I guess from a physical point of view, you know, docks have a nice, I guess I would think of them, I don't know about cakeweed if it has a big taproot, but docks certainly do. And so from a physical point of view, that might suggest that the docks are there to help grow that big taproot and help to alleviate some soil compaction or soil tightness. So maybe it's a physical problem that the soil is what those weeds are indicating. So I would maybe try and grow some other plants that also have a nice big taproot. Let some economic plants fulfill that taproot function that you can also gain an economic benefit from. So thinking about, or, you know, maybe some temporary soil opening, whatever, subsoiling or fracturing the soil to open up that compaction, but only doing that, you know, combined with, because that's, again, that's not addressing root cause, that's treating a symptom. But you could do that as a temporary alleviation, make sure that you've then got some deep rooting plants to then grow down those cracks, maybe inject some biologicals or biostimulants down there with it when the roots are actively growing, and get some roots to grow down those cracks to then open up that soil and prevent it from just recapping or not capping, solidifying. And so yeah, if you look at it from a physical point of view, maybe try and grow some similar plant habit plants, so deep rooting crops, lucerne or something else taprooted.
Yeah, excellent. So look, we're pretty much used up all of our time now. Karen, do you have anything that you'd like to add?
No, not really. I just did notice there are a couple more questions. I guess we've covered a lot of that. There's approach to powdery mildew infection in a vineyard. Well, again, I would suggest, and Joel probably would too, just increasing that biome of microdiversity through biostimulants, kelps, fulvic acids, just trying to, especially if it's in an organic approach where it's for human consumption, you don't want to be bringing in any toxic conventional products, so like fungicides and nitrate fertilizers. So it's all about just that gentle management and really bringing the biodiversity back and allowing them to do the work for you and working with nature and not against it.
Yep, excellent.
Yeah.
Very good. All right, Luke, you're pretty all right now too. Have you got anything more to add?
No, I was just going to ask Joel if he had anything he wanted to finish off with. And
yeah, I mean, I guess I would add that I think, well, on that vineyard one, you know, the huge opportunity there is your interrow, you know, get more diversity into that interrow, put some flower blends and cover crops and mixed flowering species, get that in. That's a huge opportunity to use that interrow. And yeah, as I touched on today, I would use a nutritional and biological approach. You know, yes, vines are pretty responsive to silicon as well. I would yeah, be careful of too much nitrogen. I would address nutrition, and I would do foliar sprays of compost extracts and microbials and things to provide a biological layer as well. So yeah, I would use an integrated approach of all three of those.
Yeah. Very good. All right. Okay. On behalf of, I guess, Pest and our participants too, I just want to thank you guys as panel members. Joel, thank you very much for your time and sharing your wealth of knowledge, you know, that you've sort of gained over the years. It's very much appreciated. Luke and Kieran, thank you for helping with the answering of the questions and asking Luke as well. I'll close it off now. Now, I apologise to the panellists. I'm going to try and leave us on so we can have a discussion at the end. If I flick everybody, we might have to. I don't want to be rude. So I'll end the session now, and we look forward to hosting our next webinar with Regen Farming and Pest as well. And thank you, everyone, for being involved.